A lot of families receive a report that tells them that their child has had a de novo mutation in a gene like MYT1L. And they may ask, what does that mean? What does that mean for them? What does that mean for their family? The genome is a set of instructions for parts that are needed to build different organs in the body. And we all have the same 20,000 different little parts described in the genome. For every copy of the genome, we have one that comes from mom and one that comes from dad. So you have two copies of all of the instructions. What happens sometimes is in the ovaries or in the testes, when those instructions are being copied for fertilization, there's a mistake made in one of the genes. A lot of times it doesn't matter. There's 20,000 different genes. Many of them are fine with a little bit of a misspelling in one or the other. But occasionally, if there's bad luck, you'll get a misspelling or a mutation in an important part of an important gene. And this just happens by random chance, and that's what scientists would call de novo. It's just Latin for a new event. It's nothing that the parents did. It wasn't something mom did during pregnancy. It was just random strike of lightning event that happened genetically. And it just happened to have the bad luck to happen in an important gene. So there's about a 1 in 20,000 chance, or 1 in 22,000 chance that you might get a mutation in a gene like MYT1L. And for these genes, for some of the genes, even though you have one healthy copy and one broken copy, one copy is not quite enough for totally normal brain development and function. And so the kids grow up and they can walk and they can talk, but they arrive at some of those milestones a little bit later because they're just not quite making enough of that protein that they need. And that's what a de novo mutation means. MYT1L syndrome is caused usually by de novo mutations in the MYT1L gene. So the MYT1L gene is a set of instructions to make one protein. I like to think of that protein as a little, its role in the brain is to be a switch that turns on and off other genes. So when a child has this mutation, they end up making just not quite enough of those switches. An analogy might be like a factory making cars. So the instructions for a gene are like a different piece for a car. And if you don't have enough of the instructions for a certain piece, you may not be able to have quite as much of that function as you want to. For some pieces, this will matter more, some it'll matter less. If you're missing one carpet tack in the whole car's carpet, you won't really notice, that won't change too much. But if you're missing half of the steering wheel, it might make a big difference on how easy it is for that car to drive. So these de novo mutations in genes like MYT1L are often in sort of parts that are pretty important. The car still drives, it still basically works, but it may be a little bit more challenging. So sometimes you'll hear that some mutations are what are called loss of function. Sometimes they'll also be called nonsense mutations. Other mutations might be called missense mutations. If you think of a gene as sort of a sentence that it describes the instructions to make a protein, a nonsense mutation usually means that there's an abrupt stop somewhere partway through the sentence, some interruption, some loss of function. Sometimes they'll call this a stop gain that really usually just simply prevents those instructions from being read at all. A missense mutation is more like the change is not inserting a period into the middle of a sentence like a nonsense, but rather it's like you're changing a couple of letters. So you're making some of the words not read correctly. And it's a little hard to know what those missense mutations would do. A nonsense mutation is pretty clear. You're going to lose that copy of the gene. You'll make less of that protein. But the missense, you can imagine some sentences you could read fine if a few letters were messed up. Other sentences might be very confusing if you change just a few letters in them. So for us, the missense mutations as scientists and also for our clinical colleagues are harder to interpret. We're trying to take a little bit more of a guess if we're trying to understand what those are going to do. Sometimes, in fact, they can be even more severe than the nonsense mutation, because sometimes it's worse to have a confusing sentence than no sentence at all. So you can imagine like changing a word in a sentence that would completely change the meaning of the sentence would be more risky to you understanding that sentence than having not read the sentence at all. So with the MYT1L patients, we see that roughly half of the patients have these nonsense type mutations, and that's what families might see on their genetic report. Again, loss of function, nonsense, stop gain, gene disrupting mutation. These are all sort of synonyms we use for the same thing. And about half of families will have these missense mutations. I think the families with the nonsense mutations can be pretty certain that they have definitely MYT1L syndrome. With missense, it's a lot harder. We have to sort of look at each one of those missense misspellings and really study to say, do we think this is going to cause the same syndrome? Is it Or is it going to cause something more severe, potentially? Or could it be a red herring? Could it be that this particular misspelling actually doesn't have any negative consequences, and that it's something else going on with the child? So that's where it can take extra experiments to really understand the consequence of those missense mutations.